Hi friends, today I am discussing about an introduction to biochemistry, chapter number 1. Biochemistry definition. Biochemistry defined as the branch of a science which deals with the study of chemical reaction which take place inside living organism. The term biochemistry was introduced by Carl Newberg in 1903. It is a hybrid branch of chemistry. It lies between biological and chemistry that studies complex chemical reaction, chemical structure, gives rise to life. It deals with chemical basis of life in plants and animals. It relies on basic laws of chemistry, biological and physics to explain various processes of living cells like growth, metabolism, reproduction, response towards a stimuli, etc. Next, scope of biochemistry in pharmacy. Various steps of biochemistry. First one, biochemical test. These tests help fix the specific off-life of date of expiry of drugs. Second, the off-life. This is a test done on biochemical drugs to know how long a drug is stable when kept at so and so temperature. Third, drug constitution. Chemistry gives an idea of the constitution of the drug, its chances of degradation with varying temperature, etc. How modification in medicinal chemistry helps improve efficacy, minimize the side effects. Fourth, drug storage. The storage condition required can be estimated by the biochemical test. For example, many enzymes, hormones are stored for dispensing. These get deteriorated over time due to temperature or oxidation, contamination, and also due to improper storage. Fifth, drug metabolism. It also gives an idea of how drug molecules are metabolized by many biochemical reactions in the presence of enzymes. This helps to avoid drugs which have a poor metabolism. Sixth, chances of degradation of drug. Biochemistry helps to determine stability of a drug by calculating chances of its degradation with changes in atmospheric conditions like temperature and humidity, etc. Seventh, expiry date determination. Biochemical test helps to fix expiry date of drugs. Mechanism of action. Biochemical testing provides indispensable insight into a drug mode of action which is important for molecular study. And next topic, cell and its biochemical organization. Cell. A cell is defined as a basic living structural and functional unit of an organism which is capable of carrying out the process of the life independently. Types of cell. Prokaryotic, Greek. Pro means before, karyon means nucleus. An organism without a true nucleus, but the nuclear material being scattered into the cytoplasm of the cell. These are the simplest and the smallest cells. For example, blue-green algae and E. coli. Second, eukaryotic cells. Greek, Eu means true, karyon means nucleus. An organism containing a highly developed, very complex nucleus surrounded by a nuclear envelope consisting of two membranes. Next, afterwards, the chart will come. Points to remember. Difference between prokaryotic and eukaryotic. Types of cell, always unicellular of multicellular. Here chart will come. Next, structural and functional of eukaryotic cells. Cell organelles and their function. First one, cell membrane. Intrinsic means integral proteins which are present inside the cell membrane. B. Extrinsics, peripheral proteins are present or attached to outside of the cell membrane. Here functions of a cell membrane is It holds a cell together, it secretes a waste products, it keeps out a toxic material, it prevents the loss of essential substance from the cell. Second, nucleus and nucleolus. Nucleus is the heaviest component of the cell. It lies in middle of the cell. The living matter inside the nuclear membrane is called a nucleoplasm. Functions of a nucleus. Nucleus controls all functional activities of the cell. It is the site of a DNA directed RNA synthesis which is involved in the protein synthesis. Third, mitochondria. Mitochondria are rod-like bodies. Mitochondria consist of proteins, lipids and small amount of nucleic acid. Each cell contains 50 to 2500 mitochondria. Next, functions of mitochondria. It is called as a powerhouse of a cell. All enzymes involved in a Krebs cycle are present in mitochondria. Mitochondria is responsible for the transmission of hereditary characters. Fourth, endoplasmic reticulum. Smooth endoplasmic reticulum and rough endoplasmic reticulum. Here, functions of endoplasmic reticulum enclosed spaces of endoplasmic reticulum play a role in exchange of material between the cell and the extracellular fluid. Next, second point, the exchange of a material takes place by the process of diffusion, active transport, phagocytosis or pinocytosis. Fifth, ribosomes. Ribosomes are small granules outside the membrane of the endoplasmic reticulum or are scattered in the cytoplasm. Functions, these are the site of a protein's synthesis from RNA molecules. Sixth, lysosomes. It means suicidal bags. Functions, it contains digestive enzymes for intracellular digestion. 7. Golgi bodies or apparatus. These are also called as a dictosomes. Each eukaryotic cell contains a unique stack of smooth surfaced compartments or a cisternia that make up the Golgi complex. Functions of Golgi bodies or apparatus. It is the site of an enzyme secretion and it participates in the formation of lysosomes. 8th, cytoplasm. The cellular matrix is collectively referred to as a cytosol. Its function is it helps in maintaining the osmotic pressure inside the cell. Peroxisomes. Peroxisomes are a small vesicles known as a microbodies single membrane bond organelles. Functions of peroxisomes. Oxidation of fatty acids takes place within the peroxisomes. 10. Centrioles. A number of fibrous structures present in the cytosol that give shape of the cell which also supporting cellular transport. Next, functions of cytoskeleton. Cytoskeleton provides shape and mechanical support to the cell. It allows expansion and contraction of the cell. Thank you guys.